Well, I'm very grateful to be here today to be able to talk to you about some of the stuff that we've been doing and some reflections on the field in general. And I'm going to try to follow up some of the things that that Dirk had talked about, fill in some blanks, and maybe go off in some other new directions. So I just want to show you that, in fact, I do qualify to be here because this is my sister who does not have CDG, but she's developmentally disabled and 63 years old. And just like all of you, you know, I love my sibling and your child. I mean, so you know what it's like on that side, and so do I. It's not only from a research standpoint. So I'm going to talk about an expanding universe of human glycosylation disorders. And some of this stuff you'll have seen from before. Some of it is going to be a bit new. And I want to sort of explain who I am and what I do, as opposed to the physicians that you deal with all the time, people in diagnostics. And then I want to just give you briefly an overview of how we discover glycosylation disorders. So I'm a basic scientist, and what that means is that I'm not a doctor. So I'm not medically qualified. You ask me for any kind of medical advice, and I would get in trouble if I gave you any real advice. So I'm not a doctor. My business, if you will, is to really make new discoveries. And I have been involved in glycobiology as a fundamental science for over 40 years. And the reason I can stay in business is because I managed to get research grants, oftentimes from the U.S. government, that are made specifically to my lab to make new discoveries. And fortunately, because there is so much activity in the patient connection of glycobiology and fundamental research, we can occupy a unique place. And there are very few labs that are actually doing this. Glycobiology is a sort of orphan science. If you looked in faculty rosters and you said, how many glycobiologists are in this faculty of 500? You'd more than likely get 0. There are only a couple of hundred glycobiologists, for instance, in the whole U.S. And because of that, there's not a great understanding or connection between fundamental scientists in general and physicians in training. So that's why it's so difficult to go out and find a doctor who knows something about glycosylation and about CDG. So we've been very fortunate in having not only money from the federal government, but also some generous families that have helped us out. And of course, companies who are in business to make a profit, but they also have to have a heart. They also can help out in moving our studies of CDG forward. So how do we discover glycosylation disorders? And if you get nothing else from everything I say, realize that it's you. You are the experts. You're the people who live with this every day, and we so much depend on everything that you do in cooperation with us. We will oftentimes hear from families directly. You know, as I say, our lab, and practically any lab, is a click away from your finger, and you can reach our lab. And we often hear from patients and from families directly. Some of you contact us through your physicians and we'll find out about your cases. Other times there'll be a diagnostic lab. For instance, we work a lot with the Mayo Clinic that will tell us about some interesting findings, and then we're able to make a connection that way. But again, what I want to emphasize is we rely on you and we work for you. If you're not there, I can't do fundamental research. I can't have relevance in the things that I want to do. And so for you and for me, we've got a cooperative going, and that's really important. So what have we been doing here recently? Well, if you look at this, there are probably more than 40 autosomal recessive congenital disorders of glycosylation. 4 of these are now found to be X-linked, which means it's only inherited from the mother. And in fact, we're even finding some cases of CDG where it's a spontaneous mutation. In other words, mom didn't have it, dad didn't have it, the child gets a mutation. And those are spontaneous mutations. And they're not as rare as one might think. And we know already from just hearing some of the comments that the phenotypic spectrum of CDGs are incredibly broad. And one thing I'm going to talk about near the end is a congenital disorder of deglycosylation. And you're going to say, what's he going into that for? Well, you'll see, because it relates to all the things that you do. So it's because a father's blog led us in a whole new direction. So let's think about the way all of us consider that research and discovery really works, right? So you have some basic laboratory finding over here, and somehow you do translational research. That moves into the clinic, and then beyond smaller clinical results, then you may understand something about entire populations. And eventually that results in improved public health. Well, it doesn't work that way. It works like this. Everybody's connected. You heard that before? We're all connected. And all of those discoveries that are made at every level impact everybody else. Things that will occur in population studies, we can interpret and use and come back into the laboratory and make fundamental discoveries that then cross-feed and make improved public health from connections you would never ever dream about. But having those connections, having all of your participation, is what makes the difference. So we know that medical practice and basic science sort of intertwine with each other, and they're always learning from each other. But here, the families bring in materials, if you will, your blood, your cells, your child's blood and cells, all kinds of biopsies, tests. All of that comes from you so that we can enrich basic science and in medical practice. And we owe you. We owe you. We have to have respect for you. We have to work with you as basic scientists. Oftentimes, basic scientists don't have this contact. But we need to be able to work with you. And if you send us a sample in some way or other for some kind of analysis, let's say you've just done it off the internet and we're able to work through the system and get those things and analyze them, you have every right to expect that we're going to get back to you at some point. It is not okay for us to go 6 months and never let you know what's happened. Our obligation to you, out of respect for you and what you do and what you have to live through every day, we owe you that courtesy and responsibility. If you involve us and my lab and Bobby, many of you know Bobby, you're going to hear from us because you're hanging out there looking for answers and we're trying to find them. So over the last several years, we tried, I suppose in 2010 we had about 67 patients that weren't solved. And then through a collaboration with the University of Washington Center for Mendelian Genomics, we've solved a good number of cases. Now, all of these may not mean something to you, but I want to point out one thing in particular. That's ALG1. If you go back 5 years ago, there were only a few cases. They were uniformly lethal and it had been thought about as a lethal disorder within a few years. We have found that there are many, many patients who are still alive and they're older and they don't have always a severe outcome. In fact, one mom says, well, my child can't have an ALG1 description because I know what I see on the internet and she's doing much better than that. Okay, as we learn more, we expand the spectrum, we learn more things, and we can expand what we understand and know that your child, if your child has ALG1, it's not necessarily something that is a terminal disorder. And we've solved a number of new cases in 2012 and 2013. This just gives a number, and we still have about 40 cases that are unsolved, and they're being subjected to this thing called whole exome sequencing. Now, what is that? You've heard this term before: genome sequencing, exome sequencing. And what happens is that in the past we've been able to look at single genes, and as Dirk was talking about, that takes a very long time to map out all of the potential mutations in any one gene. And there's thousands of genes. Now the technology is available for us to be able to, in a very short time, and for relatively little cost anymore, to be able to look at all the genes and find all of the mistakes. Then the problem is, how do you know that you found the right mistake? Well, if you have transferrin, if you have a transferrin indication, as Dirk was talking about, then you can begin to see, aha, this fits into the kind of pattern that we would expect, a type 1, a type 2 disorder. So we can use that information to help us narrow down the genes that may be at fault. And many of the times, that's what we've been able to do in these cases. So if we look at this interplay that I've talked about, the defective gene and the biochemical test actually go hand in hand. So what we've done before is to have the biochemical test, transferrin, then sometimes lead us to the defective gene. But the defective gene is now taking us back to indicate biochemical test so that we know specifically, yes, not only was that a mutation in the gene we thought, in fact, when we check it out using biochemical test, then we can see that, yes, after all, that was consistent with the kind of pathology that we see in the cells related to glycosylation. And again, you're leading the charge here. It's your cooperation that makes it possible for us to help you out. So, as I was saying, the diagnostic test can lead to the gene, and the gene to biochemical test. Now, what do we use? And this is sort of at the state of the art, I would say, right now. So how do we determine that a particular gene and the mistake in that gene is actually related to the pathology? Well, we use the patient's and the parent's cells. That's very common. We'll sometimes test these putative mutations in yeast cells. We'll be able to look at yeast because the glycosylation pathway is conserved in yeast and to humans, a major portion of it. So we can use those as test animals. I guess they're more like plants. I mean, they make beer and bread, so, you know, you tell me. Anyway, we can use fruit flies, you know, those pesky little things that buzz around your peaches and your bananas. Well, we use them for test systems because we can make mutations in those genes in the fruit flies and then test whether the gene mutation that we found in a patient is actually a damaging mutation. The same sort of thing with zebrafish, you know, the little fish you get in your aquarium. And we have flatworms called C. elegans. So if you use fish and flies and worms, you can come up with some picture that allows you to say, yes, this mutation that we think we found and we think is important is actually doing what we can. This part, you know, oftentimes will take a long time because we have to really be certain that we're right about this. We don't want to go tell you through official clinical labs. That's not us. Remember, we're research. But what we can do is we can guide those authentic labs to find out and prove that the mutation really is what we think it is. I'll move right along here. Just to give you a sense of, you know, this is a really big world of glycosylation, and Dr. Yakin showed something like this yesterday, but these are different glycosylation pathways, okay? And each one is in a different color, and we've primarily used this N-glycosylation pathway to show how the defects have increased. And if you go back here to 1969, that's when I sort of first started in research. I ran around Yellowstone National Park hot springs looking for bacteria that would grow in the springs, and I actually found one. And that was really cool because it turned out many, many years later that that bacterium provided an enzyme that we could then use to do all the genetic diagnosis and testing that you see that's come about from this. So planting a seed in fundamental science can sometimes pay off. You just can't tell how and when it's going to pay off. So that was in '69. Then eye cell disease was really the first glycosylation disorder. You've all heard of Manos therapy for some patients, and this is when we first treated our first patient called Max. And then here was when the human genome was sequenced, and you can see it was soon after that that we've taken off in an enormous number of glycosylation disorders. And we're approaching 100. And I calculated that a new glycosylation disorder is reported every about 2 weeks in 2013. I mean, that is a phenomenal explosion. These are the funny sugar chains you saw depicted. People stay away from glycobiology because it looks too complex. And we decided it would be easier if you could just make it all in those kinds of funny symbols. Different sugars, different shapes, different orientations can make you understand glycobiology a little easier. So that's one way to depict it. You stand it on end, and I know a lot of people sometimes think that it really looks more like just a bowl of fruity cereal. It's not. You can make various uses of that. In fact, one mom, Cindy Wren Gray, has designed jewelry that is based on this kind of structure. And you can go through the CDG family network and still buy that, I think. And so I'm going to zoom through this next part because you've seen a good bit of it. This is the same thing that Dirk was talking about, where we have an accurate balance to weigh the sugar chains. And the same sort of thing. Here is transferrin with 2 sugar chains, and it has that weight. Now, if you have a sugar chain missing, you get that peak. That's exactly the difference of the mass or the weight of that sugar chain. And if you're missing both sugar chains, you get this. So this allows us to do accurate mass measurements and directs us to whether we're dealing with a type 1 or a type 2 disorder. And for instance, you can miss individual sugars. There's one peak that's consistent with 2 sugars missing. And if you look at that one before it, that's just the mass of 1 sugar missing. So we can use that to help the diagnosis. And that guides us into place a little bit better. This is data from Kimmy O'Raymond, and I think it's really amazing. She's at the Mayo Clinic and does a lot of the diagnostics using this technique. And one of the things that she did was to look at all the different samples that she had done over time. And that amounted to about 20,000. She found about almost 900 abnormal transferrins. But look at what happens. If you break that down by different ages, okay, what you see is that the frequency of finding abnormal transferrins is highest between about 1 month and, let's say, about 13 to 2 years, less than 2 years old. That means that she was finding 10% of the samples that she got were positive. That's a good bit higher. Now, we don't know that every last one of them is a CDG. That's where you have to go in and do this final proof and to show the mutation is there. That's about 3 times higher than you get at other times. So what does that tell you? It means that you should have transferrin run early, because there may be a window where transferrin is abnormal and then it becomes normal. This is the PGM1 that Dirk was talking about. I think, Evie, you're going to speak a little bit more about this. And I don't know, perhaps Thorsten in his talk will actually mention some things about this too. I'm going to be very brief about this. But this was a big collaboration of a number of different laboratories in Europe and the US. And we all pulled together. The author list looks like the New York City phone directory. I mean, there's a lot of people in there. But we came together, we did different experiments, and we discovered altogether about 18 patients who have a very unique clinical presentation, and that abnormal transferrin that we couldn't figure out that Dirk was talking about, where you had both missing sugars and whole chains that were missing. So this was a unique identifier, and it turned out that some of these problems, such as the hepatopathy and all of the issues that we see here as a clinical presentation, some of those are due to abnormal glycosylation, but some of it is due to abnormal glucose metabolism. And this links glucose metabolism and glycosylation in a way that we hadn't really thought about before. So this gives us a new approach. And I just want to tell you about one simple test that we had done, and I think it was important. This is something Maria Stell worked out. So there's this molecule called green fluorescent protein. It's a protein from jellyfish that glows. You know, if you've seen jellyfish and they're glowing right in the ocean. So you can pull the protein out of there, and that glows. There's a reason, and there's a Nobel Prize basically behind figuring out why that protein glows and then manipulating that protein to give it different colors of glow. So what we did, or what Maria Stell did, actually was to make a green fluorescent protein variant. And what it would do is it would get to the right place within the cell called the endoplasmic reticulum. It would get in there, and then it would stay there. And she put in an artificial glycosylation site. It's not normally glycosylated. But if you put this glycosylation site at the right place, it destroys the glow. So quite simply, if you have it glycosylated here normally, it will glow. If that glycosylation site is not occupied, it will glow. If it's occupied, it doesn't glow. And this is important because we can use this as a test for finding out whether we've got the right gene identified. Because we can take your child's cells, and if it's glycosylation deficient, we'll see a glow because glycosylation is below normal. Now we can put in the right gene, and the glow goes away. So I mean, it's sort of like a light switch, turning it on and off. And then we have sophisticated instruments to measure that. So that's a very useful kind of molecule that we can design, and it's a fundamental research molecule, but we apply it to know that we're getting somewhere with your patients or your child's mutations and what we think is going to be some lessons for fundamental science. So one of the things is we're now working with this company you may have heard about and seen, met some of their people here from Agios. And they're trying to find therapies for CDG. They're going to specialize in probably PMM2 initially. Let's see how that goes. But we need to apply all the basic science tools that we developed in the lab, and we're working with them to do that. And our hope is that this kind of a cooperative is going to ultimately lead to some therapeutics. So I'm excited about this, and I'm very hopeful. Now I'm going to change gears in the last couple of minutes here. So this is a father's personal story. So I know all of you are probably looking at the internet every day. You're scouring for new evidence of what your child may have. Maybe you don't know the type. Maybe you're looking for it. And so this was a father's story about how he confronted what was going on with his child. And it was a blog, which is given down at the bottom. And, you know, you see that picture on a blog, and that catches your attention. So what are the words that go with it? I'm hunting down my son's killer. I found my son's killer. It took 3 years, but we did it. What is it? Well, it turns out the diagnosis is an N-glycanase deficiency. And you say, what is that? So sometimes when proteins become glycosylated in the endoplasmic reticulum, that's a compartment within the cell, Sometimes it still doesn't fold right. Sometimes it needs to be degraded. And what happens is that that protein is pulled back into the cytoplasm. And when it's there, it gets degraded, sort of like going through you know, one of those tree mulchers that just branches with trees and all, and eats them all up. There's an organelle within the cell that does this, but the first thing it has to do is strip off the sugar chains that it put on. And now if you can't strip off those sugar chains, you can't degrade the protein. And that's what this boy's problem was. And his dad writes this very compelling blog about their search for a diagnosis. And it's the same kind that many of you will be able to identify with. And although I can't give you details at this point, this is going to, I think, turn into a rather high-profile article in some major publication, probably in the next several months. So whether they turn it into a movie or not, that remains to be seen. But there's a lot of action going on. In any event, what was so interesting about it is that they came and contacted us because of our connection to glycosylation and because we had played a minor role in the diagnosis. Somebody else had actually found the mutation, but then we said, yeah, it looks like it is a mutation based on functional assays, glycosylation assays. And so we don't know all the basis of it yet. There was another patient that was found in Turkey, had a very similar phenotype. And this is Bertrand. And Bertrand's family visited the lab here. You know, when I say this is Bertrand, I didn't want you to think this is Bertrand. That's Ping. Ping is the guy that's working on the project full time. But 6 additional patients have been found. And let me tell you what happened on this one. A family, a German family living in India, saw the blog. And they said, gee, that looks just like our kid. And so they contacted the dad that wrote the blog, and they said, you know, Benno has exactly the same kind of symptoms that you describe in the blog. Do you think it might be possible? We've been looking for 3 years now. Do you think it might be possible that our child has the same defect? And so we were able to get cells. We were able to run our standard biochemical fundamental science test. And the bottom line is Benno does have exactly the same defect, and in fact has the same mutation. I won't go through what all this means. Just look at these numbers and where you get these scatterings of dots. Here is what normal would look like with 43. Here is what Benno had. Here is what Bertrand had. And here is what normal should be. So we know that we've tracked down his disorder. But look at the power that you have in communicating with each other. That's why you're the important people here. You're going to be able to take control to a large extent over the future of what rare disease looks like because you are committed. You are so much part of the process. You have the biggest vested interest, and we know that. And I just want to finish off here with just a couple of things. And just to show that we've done some calculations based on how frequent a group of CDG disorders might be. And I won't go through what all of this means. The red bars, and except in this case the gray bar, are the relevant ones here in the European American population and in the African American population. And the point I want to make is that the height of those bars indicates the likely frequency of mutations. And you can see that there is a big disparity between the size of some of the bars in European Americans and African Americans. And in the US, African Americans are diagnosed less often, they're screened less often, but we can see that they're going to be carrying some of the same kind of mutations in the same genes. This is our connection to patients. You're going to recognize a lot of people here. So this is our wall at the lab. These are many, but certainly not all the patients who we worked with. That's some of the people that have helped support the lab in time. And you certainly recognize Lady Gaga there. And thank you for allowing us to use that picture, Morgan. And this is the rocket himself. You've maybe heard of the Rocket Fund that helps support our lab, and you can make contributions to the lab, and it's tax deductible and all that sort of thing. And that's Rocket. And Rocket's birthday was just August 22nd. He would have been 7. So this is where we established the Rocket Fund, and people have been very generous. These are some of the people who have been in the lab. These are our collaborators. These are some of our angels who aren't with us anymore, but we still have a number of patients who are doing pretty well and still with us, and we're able to help them as much as we can. These are all the people that are doing the work. Of course, you know, we keep our surfboards pretty handy there in case we got to get out of the lab in a big hurry. And that's the Agios team. So they were out to the lab for a visit, and we hope to encourage them to keep going and nurture their interest in CDG. So, thank you.